Good morning, everyone. We'll begin with the post-Mendel theories. I'm Dr. Charu West. I'll begin the presentation. Let's begin. Uh, Talking of the post-Mendel theories, first theory comes, that is gene interaction theory. Mendel theory of inheritance, I would like to tell you, Mendel theory of inheritance was based on the idea that genes behave independently of one another. However, it was later discovered that genes do not act always independently and their expression can be influenced by other genes. You know that our body or plant systems are so complex that without gene interaction it cannot happen. So here in this gene interaction theory, it is told that genes don't act independently but also act together for a greater gene expression. Okay? So let's move forward. One another theory of later than Mendel, means post-Mendelian era, is polygenic inheritance theory. This theory, I suppose, is very important, as when we see, in accordance with agriculture, we see wheat crop. The kernel color of wheat is a polygenic nature, right? We see other traits and plants. This is polygenic nature, right? So we see that Mendel theory was based on the idea that traits were controlled by single gene, right? He was emphasizing on the fact that traits were controlled by single gene. However, many traits are actually controlled by multiple genes. The wheat example I gave you, right? The polygenic inheritance theory explains how multiple genes can work together to determine the expression of a particular trait. Now we'll move again further with the next theory. One theory is epigenetics, the theory of epigenetics, very interesting topic. You know that epigenetics refers to the study of changes in gene expression that are not caused by changes in DNA sequence. Means all the inheritance that is happening other than the DNA, means all the DNA regulatory system or DNA machineries, genes are not involved in that. If other than the DNA the inheritance is occurring, it is due to what? It is due to epigenetics, or more specifically, more precisely, you can say that it is due to DNA methylation. There is a peculiar process, that is DNA methylation, and it is, and I can tell you that it is influenced by the environmental factors. Next theory will go. That is non-Mendelian inheritance, or other than Mendel, you can say. Non-Mendelian inheritance refers to inheritance patterns that do not follow the principle of Mendelian genetics. They are incomplete dominance. Like in our schools, in the garden, there is a flower which is like snapdragon or erythronium majus, dog flower, we can say that. So its inheritance is what? Mixed, right? It's not complete. The color is in between. So that principle, that phenomena which is occurring, it is called as incomplete dominance, and it does not follow the principle of Mendel. One is codominance. Codominance, classic example is our blood groups. A, B, O, when A and I is there, they are acting together, A is dominant. So both are dominant. Means both are dominant. When both are dominant, A and B are together, AB blood group is there, right? So together they are acting, together they are dominant, so it is codominant. It is also not following the Mendel inheritance. One is sex-linked inheritance, like hemophilia disease is also a sex-linked inheritance. It is not following the Mendel principle again. So these were all examples I told you. They were post-Mendelian era, and they tried to disapprove the Mendel theories. Precisely I have discussed that. One is linkage and crossing over. You know what? This topic really fascinates me, the linkage and crossing over. Why? Because all the variation in the nature, all the we people, all the plants are a product of crossing over. Take it? If there is no crossing over or recombination, no variation would be there, no varieties would be formed. And here in today's era, as the variation is depleting, what we are doing? We are focusing on how to create novel variations. What we are doing? We are hybridization. We are using biotechnology, we are using markers, right? We are using haploid, double haploid production techniques, so that new variation is there, new combination are there. Be it like recurrent selection is a process. We are intermating things. In behind intermating, there is a picture that recombination is occurring over there. Recombination, to recombine again. So it is that in chromosomal segregate independently during meiosis. However, it was later discovered that genes located on the same chromosome are often inherited together. A phenomena is called as linkage. Mendel used to talk about the principle of independent assortment, that they assort independently genes. But later it was studied and it was found that genes which are located on the same chromosome are inherited together, and this is called as linkage. Crossing over which occur during meiosis can separate linked gene and produce new combination of alleles. It is simple. Crossing over. Again we are crossing. The genes are separating. Chromosomes are separating. Meiosis is taking place and new combinations are formed.